Well, thank you all. It's a delight to be here again. Lovely to see some familiar faces and some new ones. And our guest in conversation this evening is Siddhartha Mukherjee, who is a cancer physician, still sees patients, keeps his bedside manner brushed up, and also runs a lab looking at stem cells, both of these at Columbia University in New York. And Sid is also a writer, as some of you will know. So his book, The Emperor of All Maladies, was on Time's list of 100 most important books ever written. So no pressure there. And won the Pulitzer Prize. And also, as we were discussing just earlier, also won the Guardian First Book Prize, which I think is a good accolade for a book prize that crosses over between literature and scientific and factual books. So we're delighted to have Sid along. And, we'll chat between us. We hope you'll pay attention for a little while. There'll be time for questions in the second half. And we're not going to give you a tutorial. There's no whiteboard. We haven't brought any PowerPoint. But I did think it was worth, at least to begin with, if you don't mind, Sid, reviewing some of the basic facts about this stuff, because, just to sort of level the playing field out. And I'm going to ask what
Let me begin by asking you,
yes.
Ian Fleming or John le Carre?
Ian Fleming. You're asking me Ian Fleming or John le Carre? I'm going to answer you John le Carre. Better on love, in my view. And you?
Ian Fleming.
Very good. Well, so it begins. I'm not sure which way the house went on that one. Right, so you in this lovely book that you've written and will be signing afterwards, The Gene in Intimate History, you talk about the gene, the atom, and the byte as being, with a Y, as being the fundamental units, of the contemporary world. We'll leave bytes and atoms aside for the moment, so I think we'll return to both of them a little bit later. But can I ask what I think is probably a deceptively simple question, Sid? Can you just explain to us briefly what a gene is? Would you mind? If that's not too...
No.
We have an hour and a half.
So, a full description really merits longer than we have. In brief, the gene is a unit of hereditary information. That sounds rather abstract. Let me put it in a very concrete way. It is the unit of information that you transmit to your children. I call it a unit because it's indivisible. You can't break it up into smaller parts. It is transmitted whole, generally speaking, although it can be swapped around with its sister genes, etc. But really, it's transmitted whole to your children, and it determines some aspect of your children's physiology, or appearance, or fate, or future. There are many, many other things. There are many other properties of genes by which they're defined. But this, for this audience, for this sort of discussion, I think that's where probably the discussion should begin.
Okay. And a couple of things about that definition, which I think are already interesting. We've talked about the atom and the byte, which I said we wouldn't decompose and come back to, but we will immediately. They also have constituent parts, and we'll be talking about DNA and stuff like that. But I wonder if you can just expand briefly on this notion of its being indivisible. I think that's the word you used, of its being unitary. What do you mean by a gene being unitary, given that it's made up of these letters and the codes and things we'll talk about later?
Right. So a gene, as many of us know, is made up of, it has a chemical form. It's made up of a string of nucleotides, ACTG. And if you were to look at a gene or to read a gene, it would read like ACT, GCCT, GGCC, TT, GGG, etc., etc. If you take the entire human genome, ACTG, etc., that would be about 3 billion odd ACTGs.
Okay. In the Wellcome collection, there is a printed out version of the human genome, isn't there, with the little letters, and it's many, big volumes.
If you look at that, it would be, I estimate, around 66 full volumes of the Encyclopedia Britannica.
Okay.
So one way you could imagine it is that you're sitting in this room, and right up to the walls, every wall contains these 66 full sets, not volumes, 66 full sets of the Encyclopedia Britannica. If you were to pick out one of these encyclopedias, you would read ACT, GCCT, GGG, etc., etc. Now, from that information is obviously inscrutable to you and me for the most part.
It's just letters.
Just letters, 4 letters repeated over and over again. And yet, if you put those 4 letters into an embryonic cell, in the form of this chemical divided into 23 chromosomes. That embryonic cell, astonishingly, can build you and me with our commonalities, the fact that we have one nose and 10 fingers, and our differences, the fact that you and I don't resemble each other. You like Lecare, I like Fleming.
Fundamentally different.
Fundamental division. There is a gene for that. No, just joking. But fundamental differences between you and me, and the variation that we see in our cells. If you change 5 letters, 10 letters, 25 letters, some repeat somewhere in that 66 volume set of the 66 set of the encyclopedia, you could become a victim to a deadly disease like Huntington's disease. If you had another variation, you could fall prey to cystic fibrosis.
I still want to press you, though, on this what is a gene thing.
Oh, yeah, go back. Yes.
And I'm asking this as a humble neuroscientist, therefore, like most of you, not somebody who gets genetics, who understands this stuff. So here's the question I've got for you. When you're outside a domain, it's impossible to know which things inside the domain are contested and which things everyone agrees on.
Yes.
So if I lined up 3 geneticists, would you all agree on the definition of a gene, and that would be a dull question, and you just want to move on to the interesting stuff? Or is the question of what a gene is, the definition of a gene, is that still an interesting question for you guys? Is it something that's dynamic, that's changing over recent time? Is it contested?
And the answer is that they would 3 geneticists in the room would agree largely on what a gene is.
Okay.
They would have some disagreement about how things are evolving. There's new information coming out of a gene that is coming out over time. So they would agree that it's a unit of heredity, that it specifies information, it specifies information about physiology or anatomy or some important characteristic of information, biological information. A gene need not be a continuous stretch of DNA. 50 years ago, if you asked them, well, does a gene need to be one long stretch of DNA? Most people would say yes. We now know that's not true for most genes. In fact, they're broken up into sort of little entities, and they get stitched up together when they actually perform their function. Does one gene have to specify a single function? No, it can specify multiple functions. Does one function have to have one gene attached to it? No. One function can be the product of many genes. So there would be disagreements around these kinds of questions. But the idea that a gene specifies a unit of information, however that information is used subsequently in the physiology or the anatomy or the body of the individual, would be generally agreed upon.
And then again, I mean, the book is lovely. I enjoyed reading it enormously. It's historical in part, and we're not going to review, and we've got enough trouble getting the basic facts out. We're not going to do the history of it. But can to give you an analogy here, if we think about what an electron is, right? So many of us will remember the Bohr atom from school, which was like a sun and planets model. You had the nucleus and the electrons whizzing around it. And nowadays we think in quantum mechanics of fields of probability. So in contemporary physics, there's no way of describing an electron as a little ball that whizzes around a bigger ball. There's no balls, there's nothing. But the electron is the same electron, right? Through the history of physics in the 20th century, 21st century, there's still an electron. It's the same thing. We just described it in different terms. Has it been the same thing, this gene, through the recent history of genetics, or has our understanding of what this thing is changed in the last 50, 100 years?
So our understanding of the word gene as a unit of heredity has not changed.
Good.
That's important. Our understanding of what a gene does, how a gene acts, how it becomes functionally realized, how it intersects with other genes to produce the complexities of human anatomy and physiology, that has changed and will keep changing. The analogy that I give to is to the word encyclopedia, right? Everyone here knows what an encyclopedia is. We know exactly what an encyclopedia is. We know what an encyclopedia does.
For the younger among you, it's like Wikipedia, but it's written down. Yeah, go on.
And yet, if I were to ask you how an encyclopedia works, how an encyclopedia is updated in time, people would say, well, that's been changing. These days, we update encyclopedias in a different way than we updated encyclopedias 20 years ago. In fact, if you look under the entry encyclopedia in an encyclopedia, that keeps changing because of that reason, right?
Okay.
So there's an internal recursion about it. Genes are a little bit like that. We know what genes are at a fundamental level. We know that they transmit hereditary information from cell to cell and from parent to offspring. But the exact way that the information is encoded, what exactly it does, keeps changing. How much it engages, what it engages with keeps changing. So a simple way to think about it is, generally speaking, genes produce messages. Genes have been often likened to a kind of hard copy. They produce a soft copy of message called RNA. It's usually the only way that a gene can exert its activity. The RNA itself has some functions, but it also produces, as a consequence of other biological processes, produces a thing called a protein. Proteins are the basis are the machines of life. Our hair is a particular color because of a protein. Our noses are built of proteins and interacting with other molecules, etc. So proteins are kind of the workhorses of biological life. Genes essentially carry the code to produce messages, RNAs, which ultimately produce proteins. And this sort of information cycle allows the existence of, the creation of, the maintenance of, the building of, and the repair of human bodies.
So it's interesting. So I think we're getting the sense that the gene is, it's not complete agreement between three geneticists in a row, but you kind of agree what it is.
Kind of agree what it is,
and
exactly right.
And it's kind of meant the same thing for quite a while, let's say 100 and something years. And in fact, that 100 years, of course, predates DNA, which we've all heard of. So weirdly, the thing of which genes are made was discovered after we knew what they were. Right. If that makes sense. So tell us about how could that be? How could we have a thing which we didn't know what it was made of? And tell us about that relationship, if you like, between the DNA.
That's a very important question.
And the gene it comprises.
It's a very important question. I'm going to talk about it historically and then give an analogy. Historically speaking, and it's very, very important to understand this, the gene was visualized as an abstraction before anyone knew its chemical or physical form. Because, of course, you can see that traits or features or characteristics are moving between generations.
So you share your mum's nose or...
Exactly. Even Aristotle knew that in fact there was something about information that was moving, and he really thought about it as information. He was one of the first people. It's an astonishing idea that Aristotle thought about. There's a famous quote, I think Max Perutz said, Aristotle should have been given the Nobel Prize for the discovery of genes because he really thought about it in an informational way. He thought that there's got to be information that's moving, and he recognized that occasionally that information can go away and come back. So a grandparent could transmit the risk of a disease to a grandchild without the parent having that disease, apparently. When you think about that...
Aristotle had clocked that. That's cool.
Yeah. That's right. So if you think about that very thoughtfully, you've got to conclude that it's indivisible because it didn't go away with the mother or the parent. It's got something remained that across generations, 2 generations, and then propped up again or cropped again. This idea, this was recorded by Aristotle. So way back in history, you clearly have the idea that there's an indivisible characteristic, something, information moving, doesn't get diluted, doesn't get broken up, doesn't get sort of chopped up, and it's moving across generations. So,
by the way, just to be clear, that's so important, isn't it? Because the indivisibility of the gene, which you said, would be weird given that it's made up of all these letters. Why can't you? But the fact that it can get passed on means that there's something kind of unitary about it.
There's something that is transmitted whole. And it was, it's one of the most striking intellectuals of our time, striking scholars of our time, who failed virtually all his exams. It gives us a lot of hope. Who failed all his exams was this monk working in a small town in now the Czech Republic in Brno, named Gregor Mendel, began to study systematically this idea. What is it? How is it that this characteristic moves from one organism down one generation to the next? And Mendel's discovery, which was astonishing for its time, was that when he tracked it, it really behaved in this indivisible manner, much like others had hypothesized, that it didn't get broken up, that when he watched the movement through peas, he could almost abstract it as a particle, as something that was a physical particle. He didn't know what it was made of. He had no idea. But put yourself in Mendel's shoes for a second. And this is one of the things that I particularly love to do. Think of yourself, you're a monk in an Augustinian abbey. You believe that God made everything, and you're forced to believe, you like to believe, that this is how organisms arise. Organisms arise because some kind of seed is laid, and a kind of divine decree allows you to sort of flower into five fingers in each hand. And
we'll come on to the modification thing later.
Yeah, exactly. Yeah. 5 hands would be very nice. Yeah. So there you are. And all of a sudden, based on simple experiments on peas, you begin to realize that, in fact, information is moving. That things are not what they appear. In fact, across every generation, the reason that the pea plant acquires a purple flower versus a white flower is because something has passed on, almost like a physical particle of information. And I liken this in the book to kind of a whisper. Someone has said to the embryo, you have got to produce a pink flower or a purple flower versus a white flower. And that whisper, that idea, that atom of information then moves on to the next generation in undivided.
So, look, I'm going to do something really, really awful at this point, which is I'm going to sort of gymnastically leap over Crick, okay, Jim Watson, love to leapfrog Watson, nod at Rosalind Franklin, and take it as read that we all know the structure beautifully rendered on the screens around us, the double helix, and that's the DNA, the 4 letters that you've talked about that encode. And before we get to the kind of the questions that I think we all need to be wrestling with, and you certainly argue we need to be wrestling with in contemporary society, there are 2 little bits of recent progress which I'd like you to talk us through briefly.
Yes.
One is this gene editing thing, or CRISPR as it's known, and then I would like you to do a little bit on epigenetics risk, because I think these are 2 things which even those of us who consider ourselves to be fairly well versed in the gene thing would like a little bit more on. And then we'll come to how scared should we be, what are we going to do next, and so on. Right. So would you mind giving us those 2, if you'd agree? Thank you. So CRISPR or something, if you don't mind.
I will talk about 3 things,
please.
Actually. I'll just add to this the idea of prenatal genetic diagnosis.
That would be great.
Okay, so prenatal genetic diagnosis, CRISPR, and epigenetics.
Yeah, just so you know where we're going, guys. And once we got those down, we'll do the real
Okay,
stuff.
so you know what a gene is now approximately. It's carried in DNA, this molecule, which thankfully was printed in the right way. There's a way that people often make a mistake to make it left-handed. And this, I think, is right-handed
Good job,
DNA.
Intelligence
Good
Squared.
job.
For those of you on the podcast, check out the slide online. It's,
I know. You get a plus for that because it's a terrible error to make it go the other way around. Because molecules, like left hands and right hands, these are not superposable. So anyway, I think it's the right direction. It only coils that way. It doesn't coil this way, if that makes any sense to you. Anyway, so if you were to think about that thing that's up on the stage, actually helps as a gene. It would be, as I said, in DNA, and would have ACTG, CCTG, et cetera, coiled along its coils. Its opposite strand, the mirror image, would be a mirror image of that in a different chemical form. We don't need to worry about that. Okay. In that code would be the code for, one gene could interact with 4 other genes and code for the shape of your nose. About 7 genes, we now know, intersect with each other and approximately interact with the environment and produce height. There are at least 7 or 8. About 10 or 15 interact with each other and interact with the environment and produce the color of your skin, and so forth. Some of them are very unitary. Some of them, only one gene drives one feature or appearance. They're rare. They're generally rare. Most of the other ones sort of talk to each other and act like machines, molecular machines, that will ultimately result in anatomy, physiology, etc. Okay, so that's step number 1. We know that. Now, what does CRISPR allow you to do? CRISPR is a molecular machine borrowed from the bacterial world, which allows you to go into that DNA sequence, that long stretch of A, C, T, and G, and deliberately switch one part of the code and replace it potentially with some additional modifications, replace it with a different code. CRISPR is actually a pair of scissors. There's nothing more fancy about it. It's a pair of scissors. It's a pair of scissors that recognizes one particular sequence in the human genome and clips the human genome, or any genome, it doesn't have to be human, clips the human genome or any genome at that sequence. Now remember, when I say one particular sequence, what I'm really thinking about is go back again to your 66 full sets of the Encyclopedia Britannica. What CRISPR does is it scans all those 66 sets and says, on page 742, line 7, para 4, it reads ACGGT. I'm going to make a cut in that line. And then there are natural processes in the human cells, which will try to replace that information.
Yep.
And when you replace that information, you can switch it to a different kind of information. So in other words, you could therefore go into this 66 volume, pick out volume 7, go to page 746, pick page line 1, and say I'm going to replace ACTGCCT for ACTTCCT. That's what CRISPR allows you to do.
And just two quick questions on CRISPR, and we're going to have to go through this quite fast because there's like the future of humanity to get to. But on the how surprising is that? So for when somebody tells you a story and you're not sure whether you should go ooh or not, right? So as a geneticist, is that really new and really cool? Like if someone had told you 10 years ago that was going to be possible, would you believe them?
As geneticists, as cancer biologists, we had been dreaming of this technology for now 6 decades.
Okay.
People had been dreaming of a technology that would allow you to basically go into the human genome, recognize one part of it, and change it. And no one had imagined that anywhere in the world you would find this technology. Okay. And it was hidden. It was hidden because, and I'll tell you, it's a lovely story. I'll go through it quickly, but it's really worth knowing. It was hidden because in the bacterial world, in the microbiological world, the competition for survival is so stiff that bacteria evolved, we didn't know about it, but bacteria evolved a mechanism of delivering lethal hits to viruses that invade them. And the way they deliver these hits is to cut up the DNA of these viruses. And bacteria, because of the evolutionary pressures, it had to be cut up in one particular place. Otherwise, they would destroy themselves, their own DNA, or they would destroy the wrong DNA. Okay. So basically, bacteria evolved, unbeknownst to us, this pair of molecular scissors that would recognize, as I said, one piece of DNA, one sequence of DNA in a long stretch, and deliver a lethal cut into it. And we suddenly understood this. Scientists suddenly began to understand this and began to realize that, wait a second, if you readapt this, if you engineer or re-engineer this bacterial technology, you can suddenly produce cuts or mutations in the human genome deliberately at will.
So again, I'm trying to give us the cast of characters that we need for the drama that you argue that we feel we're playing out right now, that was not a drama from 10 years ago. Right. So we can now go in and at will, digitally as it were, edit any element of the genome.
That's right, much like you can pick out a volume of the encyclopedia, chop up one piece, replace that word with a different word.
Character
now
one.
You could say, well, let me just finish up one more thought, it's helpful. You could say, well, who cares? Well, what if that character was the difference between a normal version of the BRCA1 gene, BRCA1 gene, and the gene that causes breast cancer? If you know that if you inherit two copies of that BRCA1 gene, your chance of getting breast cancer in your lifetime was 80%. That's where it begins to matter. What if that was that volume 7, page 19, was the sequence of the cystic fibrosis gene, or a gene that carried a 100% chance of giving you Huntington's disease? All of a sudden, this business of editing the human genome, rubbing out ACTG and making it ATTG, becomes extremely consequential because you're dictating the fate of a child, an unborn child potentially, who has a risk of breast cancer or Huntington's disease, and deciding that I'm going to erase that and replace it with something new. Keep that thought.
So then pre-implantation genetic diagnosis, I think, has been around for a little while. You look at the embryo before it's implanted and you check it out, and if it's got certain things you don't want, then you don't put it in, and if it hasn't got none of the things you don't want, you do put it in, roughly. Roughly
speaking. So if you think of CRISPR as writing, because you're erasing things and replacing it with new code so you can write the human genome, you can think about pre-implantation genetic diagnosis as reading the human genome. Why does that make sense? Again, going back to our simple analogy, you have your encyclopedia, and all of a sudden you decide, you know that, again, to press the point, in volume 7, there is a code that tells you, ATTGC, that tells you that your child will have a heightened risk for breast cancer. And now, instead of using CRISPR or any other new fancy technologies, you say, what if I could detect that in a fetus? Or in before a fetus is implanted, and I just won't implant that fetus. I just read the genome, the entirety of the genome, and I say, I've just decided I'm going to decide not to implant that genome.
And implantation is kind of in the context of artificial insemination, of in vitro fertilization, but you could imagine doing that potentially for a fetus that was conceived naturally as well.
That's right,
yeah.
exactly.
Okay, cool.
So now, with pre-implantation genetic diagnosis, you can basically subtract information from your lineage. You could say that of all the sets of encyclopedias that I could potentially carry forward into the next generation, I'm going to choose only this one, and I'm going to eliminate all the other ones. And in doing so, you would eliminate certain words which were in certain sets. Does that make sense?
Yeah. Yep.
It's kind of obvious. So it's a subtractive technique. CRISPR can certainly be used to subtract. You can take information away, you can rub it away, but it's also more powerfully an additive technique. You can add information back to the genome. We've never had that ability really to do that properly before. There have been attempts to do it, so it's an additive technique. So that's CRISPR, pre-implantation genetic diagnosis.
I'm loving the bit where we get Siddhartha Mukherjee to do now epigenetics in 1.5 minutes.
Okay. Epigenetics is a layer of information. It's a very controversial topic.
Okay.
Epigenetics is a layer of information that is, so the genome, every cell in your body inherits the same genome, give or take.
Okay. So in that sense, we think of the genome for an individual as kind of being fixed. There was this kind of swapping aroundy thing with the sperm and the egg, and then you get your genome, and that's your genome, and there you go.
That's right.
Yeah.
However, a red blood cell looks totally different from a neuron. If they were to exchange their properties, your body would go mad, right? You would not have any body. So something must be telling the genome to use different genes in different contexts and allow certain genes to be activated in their neurons, certain genes to be activated in your blood cells, et cetera, et cetera. In the broadest sense, epigenetics refers to the idea that human beings, when they evolve, when cells arise from human beings and they acquire different identities, that different parts of the human genomes are activated and inactivated differently. Now, how does this happen? It happens through a variety of processes. One thing, the biggest thing, is particular proteins come and tell genes in your brain cells to turn on certain sets of genes and turn off other sets of genes. So that's probably the biggest process that happens. Other processes that are more controversial that also happen is that the genome, once it's activated certain genes and inactivated certain other genes in your brain cell versus your blood cells, it also attaches chemical tags or physical tags to the genome. And whether those impact or don't impact the way that genes are changed in your is very controversial.
And controversial to the extent that you've actually, I mean, you've got slammed for talking about this in some ways, but controversy is interesting, again, for those of us outside the domain. Is that something which people do argue about within
It's a
ethics
huge
thing?
argument.
Why so much passion?
The consequence of that is that people argue about how genes are regulated. How are genes? There's a paper today that actually even addresses this controversy. It turns out that these master regulatory proteins, so-called transcription factors, are not autonomous. We're learning about them more and more every day. Yesterday there was a paper which showed that if you eliminate those, if you just fiddle with those chemical marks, you can turn genes on and off autonomously. So there's a lot of passion about this because so far we have not been able to turn genes on and off in certain cells at will. That's why it matters. Could you change a neuron into a blood cell? It's not so easy to do, even though they have exactly the same genome. Could you turn a degenerating cell, a cell that's dying, into a cell that comes alive and lives forever? Not so easy to do. They both have the same genome. So what makes the difference between one and the other? Well, it's that layer of information about the genome. It's clear that these master regulatory proteins have a big role in this. The question is whether, these chemical tags and physical tags have any role, because in fact, we can toggle those much more easily.
Okay, so look, I want to do a quick gear change now, and in my head, the fact of your being a Pulitzer Prize-winning author is very interesting to me. And I'm someone also who's been interested in straddling the divide that we talk about, the two cultures, the art and science thing. And those of you that will have heard of the two cultures will probably know that it comes down to a bloke called C.P. Snow, who argued of these two cultures. And I think the way in our society that's thought of is about scientists and artists being different, hanging out in different clubs, talking in different ways. But C.P. Snow, it turns out, wasn't too concerned with culture in the sense that we talked about now. But he was involved in the immediate post-Second World War period in the nuclear age.
Yes.
So he was very concerned about atomic power and nuclear weapons.
Yes.
And the concern that he had was that politicians, particularly, and the public, not being scientifically literate, would make it impossible for them to make decisions about the application in that age of nuclear technology, which was, in his view, the biggest moral and ethical concern of his age and could result in the annihilation of our species. You write in your book about a letter that Einstein produced to Roosevelt where he said, we have all the pieces, this was before the bomb was made, we have all the pieces necessary to create a bomb, and suggesting that Roosevelt ought to sit up and take notice. And I wonder, how exercised are you about public knowledge of these domains, and what are the things that you think we should be worried about now resulting from these recent advances in technology that will affect us, our children, and where decisions need to be made by, is it governments, is it the UN, don't know what. Give us a couple of vignettes, if you would,
Look,
of the big issues right now.
Let me begin that with a question, and it's a thought experiment. Let me begin that with a question that I'm going to throw out to the audience, and I'm going to ask the audience to think about. Let's say you have 2 daughters. And let's say we could predict with a 10% chance, the daughters are healthy, they're growing, they're 11 and 6 years old. And let's say you could predict with a 10% fidelity in one daughter that she would develop the risk of being schizophrenic by the time she was 20 years old. They're perfectly fine now. Would it change the way you think about your 2 daughters? Right. Let's say I increase the number to 50%, the 50% chance that she would develop some other illness, lethal potential illness, debilitating illness, by the time she was 20 or 30. Would it change the way you think about your 2 daughters? Yes. Now here's the second question. If it was 10% chance, if it was that 10% chance, would you want to know? No, yes, no. Yes.
For those of you in the
Maybe.
podcast world, we've had some yeses and some nos from the audience.
Right? Yeah. If it was a 78% chance, would you want to know? Yes. Right? This is the world that we are going to enter. Our children, this will be one of, it may not be the most, it will be one of the most important questions that a future generation will be asking itself. Our generation is already asking itself. Why is it that at 75% risk, they're perfectly healthy now? Why is it at 75% risk you want to know, whereas at 10% risk you don't want to know? What is it about? I mean, the answer is kind of obvious. The risk increases, we want to be sure about the future. But what makes that difference?
Let me ask you a question. What is the science or the domain of inquiry that would answer the question you just asked, right? So you're a cancer doctor, you're a geneticist, you're a writer. But it doesn't feel to me as if any of those things, those things equip you to understand them technically, and in your book and in your life, you talk about your family history and schizophrenia and mental illness and so on. But who is it who should be looking at the question of when people want to know these things, understanding it better, helping to educate? Is that a question for sociologists? Is it a question for politicians? Is it a question for anthropologists? It's not a genetic question.
Well, most importantly, it's a question for everyone. Because if you don't participate in that question, you'll miss out on your voice in that question. It's a question that we need to ask it's a question that we need to ask children with Down syndrome. It's a question that we need to ask men and women who carry the BRCA1 gene. It's a question we need to ask people who read literature. It's a question we need to ask those who think about what suffering means. Who is us? What normalcy means? Where are we going to draw the lines? Who's going to put an arrow and say, Huntington's disease, suffering, depth of potential, grief and suffering. Here's a line, but sexual orientation, not to make silly comparisons, not part of that debate, outside that debate.
But help us, because, the question of whether you do paracetamol or ibuprofen when you've got a headache is, it's a question, but it's not one where we really feel we all ought to inform ourselves and educate and so on. I quite like to look up NHS choices and do things. So what's the fundamental difference, do you think, between these kinds of questions we're addressing?
So this is a question of paracetamol or ibuprofen is not a question of who we are. This is a question that helps us define who we are, how we will define ourselves. Who are we going to choose to be? What is going to be allowable? Who's going to allow the allowable? And if you think that this is far away, this is abstraction, who thinks about all of this? Let me remind this audience that in parts of India and China, this experiment is already unfolding. The experiment is already unfolded. There are 800 women to 1,000 men. That's on the basis of genetic diagnosis and selection. It's not someone else's problem.
This is about killing girl children.
That's right, and so it's not so. And that is a dysgenic project. It is the largest dysgenic project in human history.
What does dysgenic mean?
Dysgenic means using. It comes from the word eugenics, which is to so-called better the human population. I believe, and I think most people would believe in this country, that this is dysgenic in the sense that it uses genetic technologies to produce populations that are not ideal, that reflect a kind of a perverse desire. And if you ask, as I did in this book, if you ask the people in some of, who've studied these cultures, who've studied these areas, not across all of India, it's actually quite focused on a particular northern edge of India, they believe that bringing a girl child into the world represents for that child extraordinary suffering. Right? They feel that she would undergo extraordinary suffering, and therefore that justifies to them to limit that extraordinary suffering. Now, to this audience, you would say, that sounds patently perverse. And yet, these desires, these fantasies, have already circulated around the world more than once. They've circulated around the world in Europe, in the UK, where the term eugenics was born, and they will, I guarantee you, they will circulate around the world yet again as eugenics, as the desire to make children who do not suffer becomes personalized, a personalized variation of eugenics using a combination of, as I said before, genetic and potentially epigenetic technologies, pre-implantation genetic diagnosis, and maybe CRISPR in the future.
Now, one more thing before we come out, and you're kind of freaking everyone out a little bit here with the whole infanticide thing and the disease stuff. So let me ask you to do a little bit on the positive side, if I may, and I'm not being Pollyanna-ish here. So, you've talked about diseases that you might or might not want to know about, and you've regarded being a woman as untold suffering, which must be avoided at all costs. What are the kinds of things which we might be looking to this technology to allow us to do that are better? And
how many are there? Most of the new vaccines that you're getting are the result of genetic technologies. Not all of them, but many of the new vaccines that prevent tremendously debilitating diseases around the world are the product of new genetic technologies. The idea that you could eliminate parasitic diseases by changing the animals that carry parasitic diseases, mosquitoes, for instance, is a genetic technology. Most of us who've undergone therapy for breast cancer or for leukemia or for lymphoma are beneficiaries of genetic technology. Most of us who've undergone screening for breast cancer because there is a breast cancer gene in our family and thereby caught early cancers are the children, as it were, of genetic technologies. the idea that you can diagnose cystic fibrosis and potentially prevent decide not to have a child, even though you and your spouse may both carry one of the genes for cystic fibrosis, is the product of genetic technologies and pre-implantation genetic diagnosis. Tay-Sachs disease, the decision not to bear a child, and it's a personal decision, with Down syndrome. That's a personal decision. If you indulged in that decision, if you made that decision, you've already become part of this genetic technology. You know,
but it's very interesting though.
Raise your hands. I'm sorry,
Yeah,
this is
yeah.
an interesting question.
Please.
Raise your hands if you have thought about or actually performed an amniocentesis to look at whether your child may or may not have had Down syndrome, or know someone. This we've already entered. We have already entered for all of you. And your families, you've already entered the genetic age. It's not far away from you. You've already entered it.
It's interesting, in your book, you quote a description which I hadn't heard before, which I think is rather lovely, the chapter about Through the Looking Glass, so the Alice in Wonderland thing. And I suppose you still sound to me as if you're talking like a doctor, because when I ask you for some positive stuff, you talk about all these diseases we might not have anymore, right? Ultimately putting doctors out of business. Yes. But again, before I come, which is, you know, it's a grim prospect clearly for you, but I wonder whether there are things outside of disease and medicine which we ought to be thinking about in terms of enhancement. We jokingly referred to 5 hands before. Is this the time to be thinking about improving ourselves beyond simply the avoidance of pain and disease, in your view?
So that's an important question. It's not a great question. I've thought about the question. I find it hard for me. Say again?
I'm doing my best.
I've thought about it obviously a lot. It's hard for me to imagine, as a physician particularly, I mean, you know, that's my practice, my day-to-day life. Mm-hmm. I find it hard to imagine doing that without violating certain things that we know about human beings. Mm-hmm. I mean, in other words, what I'm trying to say is that problems we have enough. Right. That to imagine enhancement beyond problems seems to be a second-order question. Mm-hmm. It would be a kind of 2.0 question. And I find it hard to imagine that given what we know about genes, given what we fundamentally know about genes, that they don't usually act as widget makers. It's an important distinction. You know, people used to think in the 1950s, maybe, that genes were widget makers. It was like a blueprint. Mm-hmm. In other words, that you had a blueprint, much like a blueprint. You know, you read a blueprint and it says, you know, 3 centimeters above the axle, place a nut and a screw, mm-hmm. and tighten it. And when you made your device 3 centimeters above the thing, you would place your nut and a screw and you tighten it. That's a blueprint. Genes, generally speaking, are more like recipes. So in other words, and you finally bake a cake, you can't say, well, 3 centimeters above the raisin on the far left-hand corner, there was a specification for putting a second raisin, right?
Clearly not a fan of the Great British Bake Off here, but yes,
exactly.
Exactly.
Right?
Yeah.
So
yeah.
Genes interact. So in other words, the way a cake happens is that you put a whole bunch of things together in a particular temperature, in a particular context, and you end up getting a process, mm-hmm. which is ultimately results in a cake. Genes mostly behave like that, and because of that reason, you can't say, pluck the raisin out of that, and all of a sudden you'll have everything else left the same. Yeah. Whereas you can do that with a widget, right? So my fear is that if, as far as enhancement is concerned, we know so little right now about the developmental cascades of what results in a human being, that ultimately will end up screwing up the recipe in more fundamental ways. And just to finish that example, we will say, add 1/10 of the butter to the cake, as it were. But you know that if you add 1/10 of the butter, you don't get a 1/10 butter cake. Right. You get no cake. You get a collapsed mess, right? And so the analogy helps us to realize that process-oriented things that are developmental or neurodevelopmental or biodevelopmental cascades, it's generally harder to intervene on them without having many, many unintended consequences. We've seen that happen before.
One very, very final question before we come to the floor, and that is, so we're quite good, I think, in this country. Human Fertilization and Embryology Authority is, I think, I understand to be well regarded globally as a way in which we think about these things as a society and legal framework, and what's allowed and not allowed to do is regulated in that way. And I believe you have similar mechanisms in America. What you can publish in a journal, Nature and Science, is regulated by certain ethical codes as well. Not wishing to pick on China, but I mean, in China, for example, are they just pushing on and doing stuff outside of that kind of framework? Is there an international dimension to what people are and aren't playing with right now? And does that frighten you?
There's a strong international dimension. And first of all, the Chinese have their own important code. It's not that they're working like rogues outside the code. It's just that their codes are not the same as ours. They don't hold, I mean, I talk about it in the book, if they don't hold the same sanctity they don't have the same idea of sanctity. They're different ideas. Not that they don't think the sanctity at all. It's that they don't have a different idea of sanctity of the embryo. And therefore, certain things are intervenable and certain things are not intervenable. The Chinese experiment that was done, which tried to alter human genome in about 70 embryos, was actually done relatively carefully. These embryos were picked, and they were picked for the fact that they wouldn't normally undergo full development because of certain abnormalities. So it was actually carefully done. That said, now that the proof of principle has been done, the next natural step would be, let's try it in a real embryo with potentially a real problem. So let's take an embryo potentially with a terrifying disease, like one of these diseases that causes hemophilia, right? And why don't we correct that gene? So it's not as if there is no code operational, it's that there are different codes operational, and they depend on the religious and scientific cultures of these individual places. So we've got to have a common code, otherwise this will become an arms race. People in the United States are already worried that if we give this away to places like China and Korea and India and other places where there are different moral codes that are acceptable, then we will lose this arms race, and it's too important to give away. So I think we need an international dialogue. If we need an international dialogue, we need the vocabulary. We need to understand why it's important. If you need the vocabulary, you need to read, don't read my book, but read the several books about it so that you can then come to the stage and say, I understand where we are. I've put a stake down. I know what a gene is. I know how easy or complicated it is to alter them. I know what it means to alter them. I know the history. I know what has happened, the fantasies of altering genes in the past. I know those fantasies are likely to be repeated. Therefore, I conclude that these are the boundaries that I will set for us and for the world.
I was in the States during the race where George W. Bush was running for president, and he came out against stem cell research.
Yes.
And I've been campaigning for years that science should be part of culture, should be part of politics. We should discuss it. I remember complaining to a friend of mine who was the political correspondent of the ABC about the fact that George W. Bush was against stem cell research. And he said, dude, you can't have it both ways, right? If you want it to be part of politics, then they may be ruling in a way that you don't agree with.
Yes.
Right? So like the fact that it's part of politics means that it can be a political decision that you don't agree with. And are you willing to give up control to the politicians of the domain?
That's right, but the important thing is, of course, politicians are us. They're not them. The minute we think of themselves as them and us, of course, we lose the fundamental quality of what it means to have representation.
Very good. Politicians or otherwise, folks. Questions, comments? Now, we'll do it with microphones. Please make yourselves visible in some way by waving something. We'll take them in batches, folks, and also just say them. It's not necessarily the case that we will answer in detail everything that's raised. It's as much to hear from you as to raise your concerns. Please, if you have a consultation, the doctor is not in. So please do not bring your personal or family medical problems to the fore at this stage. Yes, there's a wave over there. Let's try that. Sir.
Hello.
Yep, you're on.
Is it working?
Yep.
Dr. Mukherjee, this is perhaps tangentially related to the talk, but as soon-to-be parents, my wife and I know of blood cord stem cell storage companies, private companies that store stem cells from the blood cord. Can you shed some light on this? I mean, we have done our research and read, but what do you think of collecting stem cells of babies from the blood cord?
Okay, I'm gonna get you to buffer these. I'll try and make a note. So blood cord stem cell buffering. Over there. Yes. Sir.
Good evening, Doctor. I just wanted to say, in the start of your book, you tell a very emotive tale about your cousin, Moni. And for me, that felt like we were going beyond the empirical realm of the gene and going towards the metaphysics of what it can actually do to us as a society. So do you believe with this, fast-paced scientific discoveries that are happening, have we as, moral thinkers and philosophers and ethicists, have we keeping up with the discoveries that are happening? And if we don't, will it have negative implications on the future? Thank you.
Okay, that's a nice question. Well, shall we try one more? If anyone up in that, on that side? Yeah, go on. Let's get some ladies in next. Sir, please. Yep.
In your personal opinion, should we wipe out the Anopheles mosquito?
Ooh. Is that what's that one? That's the one that's doing what?
Malaria and
that one.
variety of other.
Yeah. Also, and wasps. Can we get rid of wasps as well? That would be great. I think that would be good. Yeah, I never know. Okay, so you can take all or any of those in any order you like.
Well, let me talk about the mosquito first. I'll keep the stem cell question for later.
And the wasps,
I'll answer the metaphysics question a little bit. So, mosquito is an interesting question. Actually, I have a conversation with, of all people, Bill Gates, which is coming out. You can find it in YouTube, maybe early next month. He reviewed the gene. And the mosquitoes, so I read a lot to prep for this conversation. And as you know, I actually don't go in great
conversation with Bill Gates. Exactly.
I don't go into great detail about GMOs in this book because it would really require a new book, a separate book. And I think it's an important book to write. Someone else should write it. But mosquitoes are, I'm told, and as I understand it, particularly the Anopheles, is a very unique situation where when ecologists and other entomologists, insect people, have looked to ask the question, is the mosquito useful? People have come up with the answer, no, not really. You can't find any use for the mosquito. It's not a keystone species. It's not required for the survival of another species. It is an unusual situation where this animal has really learned to parasitize, or I should say feed on mammals, and carry forward diseases that are very devastating to humans and other animals. So in the unusual case, and I think it's a very unusual case, in the unusual case of the Anopheles mosquito, or perhaps even the Zika-carrying Aedes aegypti, it would be the rare circumstance where you could make the case for genetically modifying organisms and not have a unintended impact on ecology so widespread that we would never be able to recover it.
You're familiar with The Onion, the satirical newspaper, has a brilliant headline, scientists announce breakthrough that partially solves problem created by previously announced breakthrough.
Right. And we know those parts, I actually talk about them in the book a little bit. No one thought that introducing, what harm would there be for a few rabbits to run around Australia? Yeah. Would we really care? It would be nice food for some people once in a while, et cetera, et cetera. I can give you 1,000 examples. Now, this has been studied. I would say that there is a case to be made, and the devastation is enormous. Malaria is enormous. As you know, there is a plan and one experiment to do this for Aedes aegypti in the Caribbean and subsequently in the United States. So we'll watch to see what happens.
Just to be clear, with the genetics of this one, so as it were, so we could do it with like really powerful, bug spray or swatters or something. But is there a genetic solution to that? And does this come under the kind of things that give you pause?
That's correct.
Worried about what that would
look like? In fact, there's some debate, a lot of controversy about this. In fact, I had a conversation with people who are reading this. Until recently, there was not a lot of evidence that bug sprays, et cetera, spraying worked at all. Okay. In fact, most of those were negative. So if we've run out of those kinds of solutions, the genetic solution would be to equip maybe the female or the male mosquito, it doesn't matter depending on how you want to do it, with some gene that basically renders their offspring sterile. So that you would basically have a dead lineage. Every time that mosquito reproduces, you'd release these mosquitoes. They're marked, as you may know, they're marked with a jellyfish gene that makes them glow red. They're not visible normally, but under the microscope you can figure out as the population is changing. You could trap them and watch the population change. You could equip these mosquitoes with a little extra drive so that they reproduce a little bit faster and so forth. People have very, very thoughtful people have thought about doing exactly this to eradicate malaria. Whether it works or not is a question. What its unintended consequences are is a question.
Right.
Malaria is a terrible disease. I had dengue. I was in bed for 3 months. It's a terrible disease. So these are not to be taken lightly.
You want to do cord blood or you want to do metaphysics?
I'll do metaphysics just
All right,
because
just
I'll
do
come
quick,
back to
quick
cord
bit
blood.
on
Sorry.
metaphysics and then we'll do cord blood. Yeah.
The metaphysical
So the
question.
question is roughly, so it's about, so the book, it contains stories, and you refer to them obliquely in your remarks so far, about your own family. And I think, one of the nice things about the story, this whole story, is that it does relate to all of us as individuals. You
Yes, it's
begin the book
everyone's
and
story.
throughout, you're not there as a cancer doctor, you're not there as a geneticist. And I suppose in a sense the issues that you raise do have resonance for all of us in terms of how we think about ourselves, how we think about our parents, how we think about our children, how we think about society, children. What, is that what excites you about the domain? What do you think we need to be doing?
Well, I mean, if you look as someone who's thought about this, particularly with my previous work, etc. If you look, it's almost always that that metaphysics and technology in particular are in a kind of cycle with each other. Okay. One doesn't even exist without the other. The questions that were raised once we began to have instruments that resemble human intelligence are unique questions to that moment. You could have thought about them beforehand, but they would be abstractions. But all of a sudden, when we have that moment, you suddenly begin to realize that this question has an important consequential metaphysical dimension. And so it is with genes. People have thought about for a while, kind of abstractly, what it would mean to influence the human genome. People have written about it. Great writers have written about it. Aldous Huxley, George Orwell, others have thought about this idea. But now, all of a sudden, it becomes a very concrete reality. And so therefore, the metaphysical challenges, this philosophical challenges, what do we do? Who do we define as normal? Who is there going to be an appointed body of individuals that says, that these forms of prenatal genetic diagnosis and or implantation are allowed, those are not allowed, these, etc. Who's going to be in that? Are you going to give, as I said before, the voice to someone who has a potentially lethal genetic disease? Is that person going to be represented? And is that person going to be represented adequately in that voice? So these questions, I think, are becoming more urgent as we discover the frontiers of the technology. I'm afraid we don't have answers to these questions right now. The book tries to give an answer, by the way. Yeah. I can talk about that in a second.
So you want to move on to the next batch?
Sure.
Okay, cool. There's one up there, I believe. Am I right in thinking that? You were waving just randomly? All right.
I can't see. So.
Yeah. I can. So there's.
we got one here.
Yeah, good. I thought he was raving for a reason. Yeah.
Hi, thank you for your talk so far. It's been very illuminating. You spoke about eugenics for a little while, but I wanted to ask you what your opinion is, where you draw the line, because some genes, such as sickle cell anemia, which is an awful disease that affects a lot of people in particularly Africa, also confers a protection against malarias, and as we've been talking about, that's a big problem. And so where do you draw the line with which genes you would go to protect against and which you wouldn't? And how would you then go about informing or recommending a course of treatment to a patient?
Yeah,
very nice.
The story of sickle cell disease, and actually it's in the book, is an informative story, because you could ask the question, well, why on earth do we have sickle cell anemia? And part of the answer, which has come out from evolutionary scientists, is that it turns out, we think now, that particularly having one allele or one gene or carrying one variant of that sickle cell gene may protect you from malaria. Whereas having, of course, it also sets your children up for having the risk of the disease. So similarly, people have asked the question, well, why does a cystic fibrosis mutation even exist in human populations? Well, again, in that case, there's a thought that having one allele of that cystic fibrosis gene, when you have two of them, of course, you have the disease, but having one protects against lethal infections of cholera and other kinds of abdominal.
But listen, in answering this question, I do think the question has posed it in an interesting way. So she said, you decide, and then she referred to you as a clinician.
Yeah.
What would you recommend as a course of treatment? But I think this is tricky, right? So what you've done is you've answered with a Just So story, Roger Kipling, about two Just So stories in a row. And depending on how convincing we find those Just So stories about whether there's a good mutation or a bad mutation, we might consider excising it or not. Is this really a satisfactory procedure for deciding the things that we are or aren't going to do with this technology?
No, I think the more satisfactory procedure for dealing with this technology is to ask the question, do we need to make a germline? So I have to explain two words that are relevant here. There are two ways to alter genes. One way to alter genes is to alter them such that you change them in the sperm and eggs, and therefore it becomes permanently imprinted in the human genome. And every generation thereby to come in the future will carry that genetic change. That's called germline gene changes, whatever it might be. Another way to change the human genome is to say, okay, I'm not going to do this in the germline, I'm only going to affect the cells that are affected. So in sickle cell, it would just be the blood cells that are affected. So in fact, if you were to think about it, for diseases like sickle cell, the much, much simpler solution, technically, technologically, and perhaps even ethically, is to say that we're going to make the change in the blood cells alone. You have the luxury of doing that in that particular disease, in sickle cell anemia, and I'm going to concentrate my efforts. I'm going to largely concentrate my efforts on changing blood cells so that the patient with sickle cell anemia now carries sickle cell anemia, as it were, in his or her body, except in the cells that are most severely affected, and that's in blood. Now, you could say, well, that's a weaselly solution.
Well, it's weaselly in the sense that you're limiting the consequences of the decision, but you're not addressing the question of which things you would do and which things you wouldn't do in the first place.
That's right. But it's important to recognize that these weaselly solutions are probably the most practical solutions. So that's what we would do first. That's what I think we should do first. Right. The second question, should we eliminate the sickle cell gene, the mutant sickle cell gene, completely from the human genome? From the human genome? I don't know the answer. I don't know. In the book, what I do describe, what we know historically, is that there's a kind of triangle, as it were, of forces that have allowed us to make these decisions. And it's important to note the realms of that triangle. One is that the disease should cause extraordinary suffering in being inherited through the germline. Mm-hmm. Now, you could say, well, your definition of extraordinary suffering is not the same as mine, and absolutely it's not the same as mine. It's changing all the time. But at least the standard is that the suffering should be extraordinary, that there should be something to tell us that what the consequence of having that gene causes a massive disruption of what we would consider a normal human life. Yep. So that's one.
You don't mean being born female, right?
That's right. Yep. So number two, that we understand as clearly as we can possibly can, that actually changing that gene will have a one-to-one relationship with what ultimately happens and not affect the whole, right? So that we don't have all these potentially unintended consequences of eliminating malaria and finding that something awful has happened as a consequence of that. So we have, and one way of saying this is also to say that that the penetrance, our understanding that the gene will actually exert its effects in a one-to-one manner is very high, number two. And number three is that we know from the history, now I'm talking more historically as opposed to biologically, that we know that mandating any of these choices is a terrible idea, particularly mandating it from the state. When states mandate this, they make terrible choices. So it's generally better to leave these decisions to individuals. They may choose or not choose to have a child with a potentially devastating medical condition. They may choose to have that child. But mandating it at any point of time is so this is a generally safe triangle. With sickle cell disease, we have other solutions. We have other potential. So it doesn't really fit in that triangle, as it were.
Very good. Yeah, well, somewhere there. I'll take a batch more, so if there are any others, we'll go. Yes, sir. Or whoever it is. Where are you? Oh, hello.
So do you think doctors should be the gatekeepers to the human genome? Because there are commercial human genome kits where you can kind of carry out a test on yourself and it gives you results which may not be accurate. But given that it is your own information, shouldn't you have access to it without someone else?
Yeah, should doctors be gatekeepers like that? Over there.
Hi, Dr. Mukherjee. I was just wondering where you think the future of genetics is going in terms of microbiology, because I know that so far it's seen as less powerful to actually go ahead and kill bacteria, but more powerful to inactivate genes that cause virulence. And I was just wondering if you think this is something that the bacteria would eventually evade, or if there's a future for it.
So yeah, so you're going to put the microbiologists out of business. and I'm delighted to say another view from the gods, yes.
All right, so basically you've been talking about how CRISPR and using the genome can be used to eradicate disease, but what do you think about producing like designer babies? If a consumer wants to create their child to have, say, a faster metabolism so they can't be as fat, you know, or change their eye color or hair color, should that be allowed, or should you kind of keep a lock on that? Because then it could be a slippery slope and who knows what's happening.
Right. So should we park microbiology, or we want to do that one quickly?
Let's come to microbiology
Okay,
later,
cool.
because these
So
two
yeah,
questions
these two are kind of related. So
related.
should you guys be the gatekeepers? Should, you know, who here has sent off for a genetic test out of interest? Who, if you've sent off for 23andMe or one of them online, other products do exist, put your hands up. So wow, we got like less than 10%. Have you?
10%.
Well, you didn't need to because you're in a...
Actually, I decided not to.
Okay, cool. All right. So should we have access to it, and should we be modifying?
So the quick answer is that right now, these technologies, especially the sequencing technology, is easy. That's the easy part. Reading ACT, GC, TTC is very easy. It's
How much does it cost right now? I mean, in your lab, if you want to send one
so
off?
if I want to do the entire human, the active part of the human genome, it's about $1,200.
$1,200.
Yeah,
Nice.
23andMe does some subparts of it
Yep.
for even cheaper, et cetera, et cetera. You give or take.
Yeah.
The first genome, by the way, cost about $3 billion.
Yep.
The full genome.
Yeah.
Just to remind.
Yeah,
that.
that's
the
even
full
faster
genome.
than Moore's law, isn't it, in terms
Exactly
of the
right.
reduction?
Exactly
Yeah.
right. So,
Okay.
okay, so that's the background. The problem is not that. The problem is not sequencing. Getting sequence is easy. The analogy is, you know, if you put your child and you put them in front of a book, right? And at age 3, that child can say, that's a letter A, that's a letter C, that's a letter Q, et cetera, et cetera, et cetera. They would have not one bit of understanding of what those sequence of letters meant. They wouldn't know, therefore, what the plot of the book was, what the moral of the story is, what the lesson is, and what the historical context of interpreting that lesson is. So we are sort of like the children who are reading in our case, A, C, T, and G. And occasionally we can make out this combination of letters means that. But that our vocabulary for that has about 6 or 7 words. And there's a big, you know, there's Richard III sitting in front of us. So you can read the word that, you can read the word king, you can read the word, you know, clown. And now you're asked to say to yourself, do you understand the significance of this play? Right? That's where most of the technology is sitting right now. It's at the toolkit stage. So for me, in particular, and I have a great incentive to do this. I wanted to know, is there a risk for me or for my children to have an increased risk of a mental illness? It just wasn't there. We don't know what that will be, and chances are it will be, when we come to it, it'll be probabilistic. It'll be that kind of situation, 10% risk, 12% risk. It has to do something with the environment. Some component of the environment has to be sort of factored in as well. So
you advise people not to use these? I mean, you know, whatever, you're not going to tell me what to do, but you think it should still be coming through a medical gatekeeper?
I think it should be, I think that it should be done preferably on a trial basis. It should be done in a way that information is recorded and sent back, so in a way it's useful. It doesn't become a kind of free-for-all. I think the information is helpful when you do have a notable family history of a disease. That's when it becomes useful. But in my case, with most chronic diseases, where you know that there's not one gene and the genes haven't been identified, for me right now, is not useful at this point of time.
And I just about to say, I mean, shouldn't, you know, can't we have a blue-eyed one, darling? Go on, let's just
exactly
do a little, go on, just for a change.
So let me tell you where I think the trajectory or the vector of my fear is. I think the first thing that will happen is height. I think height, because of the nature of the genetics, I think the first thing that people will try to do through prenatal genetic diagnosis, I'm afraid, will be to parse out the 6 or 7 or 8 genes. We know that there are about 8 or 9 of them, which have strong influences on height. And they will try to influence them. And I suspect that they will try to influence them in cultures where there's a premium placed on taller individuals.
Okay, so that's a speculation, but this makes you worried, or this is just you're going to invest in the company that cracks the height genes?
No, I think it makes me, if I were Napoleon, I'd be very worried. I would cease to exist.
Yeah.
It makes me very worried. It makes me very worried because it creates a culture of normalizing. It creates a culture of homogenizing. It perverts the idea of human potential. It says that the human potential lives in certain inherited features, which we have spent a vast amount of our culture trying to resist. We have spent decades of our culture trying to precisely negate and resist that idea that your potential lives infinitely in some kind of aspect of your physical appearance within bounds. I think it would be a very worrisome phenomenon, and this is why you, whether you're tall or short, whether you are female or male, whether, wherever you come from, need to be part of that conversation. Because if you're not, then someone will say, look, we have this company, what wrong are we doing? What's a little bit of height between friends?
But essentially, you come across quite, in contemporary politics, quite nanny-statey, right? I mean, you do think that this stuff should be regulated and controlled or moderated in some central way, right? I mean, that's what
if that's
you're
the
suggesting.
definition of a nanny state, then yes. If it's the definition of a nanny state that we should not be making viruses carrying human oncogenes, yes, that's the nanny state that I want. Absolutely. But that is not the standard definition of a nanny state. In other words, if the definition of a nanny state is to help us police and understand the outer limits of dangerous biological interventions that have the capacity to erode our culture that we have spent so much time investing in creating, sure, that's the nanny state I want.
Very good. Any more from any more?
Maybe one last question?
No, a little more time.
Do we have time? I don't know.
Yep. Oh.
Stem cell banking. Yes,
They
stem
want
cell
you to
banking.
answer the stem cell banking question.
So,
you said, go on.
it's a slightly difficult question to ask because we don't know what the potential of these stem cells is for the future.
So give us, for those of us that don't know, why would anyone want to consider banking the embryonic stem cells from the...
So they're not embryonic stem cells
sorry,
right now.
forgive me. So
they're
what's the
blood
question?
stem cells. They're cord blood stem cells that you can bank. Potentially you could make other kinds of stem cells out of them. They have a relatively limited use right now. They're used in transplantation if the child would get on a rare occasion, would get a leukemia. They would get a blood stem cell cancer. Occasionally, if they would have a cancer that would require severe chemotherapy. If you feel as if, you are at that level of risk aversion for your child, I would say do that banking. But realize that right now, the technology really is limited to the use of these cells for the transplantation of children that have life-threatening leukemic diseases, which is a small number of cases. Very, very rare disease in children.
Very good.
It's a
I
disease
think
that I treat,
we're in the middle
so I know a
there,
lot about
and then
it.
we'll come to the front. Yes, ma'am.
So good evening. My question is sort of medical, half ethical. We have a friend who is in the middle of a Huntington's discussion.
Yes.
Now, obviously that is binary. You either do or don't.
What do you mean, what is binary? Are they being tested or not tested?
No, if you test, you either have the gene or you don't.
Yes, okay.
So the problem is, if they are tested, they then have to report it from an insurance point of view.
Okay.
So there is no motivation to actually ask the question. Now, at what point, in terms of the genetic research, do I not want to know, because it is going to potentially affect my access to medical care later on in life?
That's a very important question.
Good.
And
I'm going to, we'll go straight on that one. Yeah, go. Correct.
So right now, as you know, in the United States, if you get tested outside, if you're not asked to be tested, your decision to reveal that information is a private decision. In fact, even if you were to be tested for BRCA1 or BRCA1, etc., your decision not to reveal that information is a private decision. And if we are to continue our current insurance system, and there's a big if there, we can talk about that separately, it seems to me that that decision should be left private. Otherwise, we will divide the world into different medical classes, and the whole point of insurance will begin to collapse. The whole point of insurance is to combine risk in a way that allows you to combine risk before information is available, combine risk or pool risk such that if you were people who've read Rawls would be familiar with this, that the whole point of insurance occurs before the veil or while the veil of ignorance still exists.
So if we all knew when we were going to die, there would be no market for life insurance.
That's right. If we all knew exactly when we were going to die, we would not have any mechanism of having life insurance because, of course, you would know when, and the premiums would change based on that. Now, the other question that you could be asking, and you have asked implicitly in your question is, does the new form of understanding genetics force us to rethink insurance? Should the person with Huntington's disease be pooled with someone else who has another lethal disease or someone else who has Huntington's disease? That's a decision that genetics will never solve for you. It will only give you information about what the relative life expectancy or risk is for an individual who has that illness or disease. The questions of how we choose to insure each other is a question that comes from social contract building. And social contract building is a decision that is a political and humanistic decision. We have decided to build social contracts around things like insurance because we believe at a fundamental level that all human beings come with a level playing ground of risk, and that should one acquire greater risk, then the pool of monies that enter that pool should be deployed to help you through that period of risk.
I'm loving the idea of life insurance as being the bit. We know we're getting metaphysical when it comes down to life insurance.
Sure. So the question that I would pull out is that as we get deeper and deeper medical information, and this is actually a very relevant question, the gene ApoE4, for instance, has powerful risks associated with it for disease. Should the insurance companies be asked to then divide human beings into two classes of those that carry the gene and don't carry the gene and therefore have two different premiums attached to it? That would overturn the way we've thought about insurance for the history of insurance.
Can I just ask you a really trivial question which just occurs to me? What was the name of the gene that you just said?
ApoE4.
How are these names chosen? What do they even mean? Is there some committee somewhere? Because like with hurricanes, there's this, they've got the Hurricane George and Matthew about to hit Haiti. Who names these things and what do they even mean?
Well, biologists name them based on their function.
Is there a committee?
There is no committee. There is no committee right now. You get to name them if you discover them.
Have you named any?
Have I named a gene? No, I have renamed. I have renamed a gene.
Okay.
We have found.
The lab
has found novel functions to genes that already had names, which would belie their names. In fact, they're the opposite of their names.
Right. Till now you've been called John, but I just think Matthew is a much better name for you.
Exactly, yeah. Okay, yeah. Generally, of course, we try to stick to convention and try to respect the first person who named the gene, who may or may not have been right. Okay. The genes are often named for absurd reasons. BRCA, BRCA1 or BRCA1 was named because of its association with breast cancer. Breast cancer.
So BRCA is short for, I never knew that. Okay, cool.
Other genes are named, Huntington's disease is named because it is related to that Huntington's disease gene. The gene that causes, that metabolizes cholesterol has a funny name, HMG-CoA.
Funny and peculiar or funny ha-ha?
No, not funny ha-ha.
Okay,
funny and peculiar.
Are there any, the genetics jokes? All right.
Yes.
Ma'am. Yes.
Hi, and thanks for your talk. So, as you said, in the US, genetic screening is getting cheaper, and then you have all the direct-to-consumer genetic testing companies. So you send your saliva, they send you all these
23andMe.
all the serious diseases, cancer predisposition, and so on. So is this information overwhelming or empowering? And also, which are the implications when a private company technically owns your genetic information? Going back to the insurance companies, well, in Canada, there is no law to prevent genetic discrimination. So what happens when a private company owns it?
Yeah.
So,
I should say in the history, I mean, just to give the historical context, this question has been very alive from the beginning of the Human Genome Project, hasn't it? When there was a battle around that. Does this give you pause?
So, who owns that information? The answer is, and what happens when private companies own the information? The answer, of course, is that companies like 23andMe have been very, with great alacrity, have told their consumers that the information will never be released. that they are under no obligation whatsoever to release it to an insurance. So in other words, you're an insurance company, you're Blue Cross Blue Shield, you cannot call up 23andMe and say, can you tell me whether you happen to have a mutation in BRCA1 or BRCA2. You cannot do that. 23andMe has no obligation. And if it were to be asked, it will fight a legal battle, and it will win that legal battle, as far as we can
Never
tell.
mind hacking iCloud for nude photos. Let's hack the gene companies and work out what...
So that has actually been raised. That question has been raised. What if they get hacked?
Yeah.
And now start releasing information, and that information becomes public. It's an important concern. I mean, you're hacking Yahoo. You can certainly hack any of the servers. There have been now all sorts of new mechanisms. Most of these private, the consumer companies have new mechanisms to anonymize, and so that you can't just decode and attach it. You know, as you know, these can be broken.
Like what they do with credit, yeah.
Yeah, they can be broken and have been broken by other companies in the past. It's a new moral sphere that we don't know how to deal with. Look, in principle, do I have a problem with private companies owning genetic information? No. If it's secure, if it's anonymized, if it's carefully curated, no. The question is that this information right now is still in a readability phase. We're still learning how to read it. For the most part, you know, you get information about certain disease risks. You get very good information about ancestry. If you're interested in ancestry, that's a great use. If you're dying to know what your ancestry is, that's a great use. In terms of predicting future risk of disease from your genome, even the most advanced genetic centers are struggling to do this for reasons we told you already. Number one is usually most human diseases are not the product of one gene but many genes. And number two, there's a rich role of environment and chance interacting or intersecting with genes that governs these kinds of decisions ultimately. So I think the chance that from a privately owned company that you will suddenly get information that's valuable to your future health, not your ancestry, still remains in its real infancy.
Come back in 10 years and tell us more.
Come back in 10 years, and you know, it depends on data, right? Depends on data. If 1 million genomes were collected, and you could have the capacity to now look through, it's a numbers problem. You know, more means more. Right. The more you collect, the more you learn. The more you collect, the more you learn. So there's a little bit of a feed-forward quality to this. So if 1 million genomes were to be pooled, I think we would start getting more information, real information about risk.
Very good. So look, I'm delighted to say that Sid's going to be signing books in the foyer outside afterwards. My name is Daniel Glazer. Please do look us up at Science Gallery London. And in the meantime, join with me in thanking our speaker, Siddhartha Mukherjee, very much.
Thank you. Thanks a lot. And thank you, Daniel. Thank you.